Hello students, Manjunath here. In this video, we are going to discuss about the questions on the chapter molecular basis of inheritance. Okay, in this particular video, I will discuss the questions bit by bit. That means concept-wise questions will be discussed, right? So coming to the first question, that is, the scientists involved in discovery of DNA as a chemical basis of heredity were, okay, option A, Hershey and Chase. Option B, Griffith and Avery. Option C, Avery, MacLeod and McCarthy. Option D, Watson and Crick. So, according to the question, what they have asked is who have done the biochemical characterization, okay, who have done the biochemical characterization of Griffith experiment, okay, or else who have given the DNA as a chemical basis of heredity. So the option here is the Avery, MacLeod and McCarthy. Okay, I'll show you. Okay, the option here which is correct is Avery, MacLeod and McCarthy because these three scientists have done the biochemical characterization of the Griffith experiment. Okay, coming to the next one. Blender's experiment to prove DNA as genetic material was performed by. Students, do remember Blender's experiment is nothing but what? The experiment which was done by, okay, Michelson and Stahl. Okay, the Blender's experiment. It is also called as Blender's experiment. Michelson and Stahl's experiment is also known as Blender's experiment, which was the experiment that proved DNA replication is semi-conservative. So the options here are Hershey and Chase experiment, Michelson and Stahl, Watson and Crick, and the fourth one is Rosalind Franklin. So the right option here is option B, that is Michelson and Stahl. Okay, coming to the third question. The genetic information is carried out by long chain molecule made up of. Okay, the genetic information is carried out by long chain molecule made up of dash. Option A, amino acids. Option B, enzymes. Option C, nucleotides. Option D, histone proteins. Okay, what they have asked here in this question is genetic information carried out. That means the genetic information, okay, the carrier of genetic information, nothing but what? The genetic material, okay. So the genetic material will be made up of dash. So genetic material, as you already know, it may be DNA or RNA, and the basic unit of DNA and RNA, or the building blocks of DNA and RNA, is what? The nucleotides, right? So here the option C is proper one, that is nucleotides, the building blocks of genetic material, that is nucleotides. And you already know that nucleotides are the molecules that are made up of three units, okay, phosphate group, okay, then pentose sugar and nitrogenous base. Okay, coming to the next question. DNA has alternate grooves, okay, which are they? One major groove and one minor groove, two major groove, one minor groove, one major groove and one minor groove, two minor groove, sorry, and last one is two major groove and two minor groove. Okay, always do remember DNA will have alternative grooves. Okay, that is correct. But how many minor groove and major grooves will be there? Alternative, only one major groove and only one minor groove will be there. Okay, as you have already studied. And what is the size of major groove? It will be around 1.8 angstrom, sorry, 1.8 nanometer or else 18 angstrom, right? What about minor groove? 2 angstrom or else 0.2 nanometer, right? So about that we had discussed already in the previous part. Okay, in the theory part we have discussed about the different size and different lengths of what? The parts of the DNA. Coming to the next question. How many coils that DNA makes around one histone octamer core? Okay, how many coils that DNA makes. That means if you consider this as histone octamer core, okay, around this histone octamer core, the DNA wraps how many times? Okay, as we have already discussed in the nucleosome, the DNA wraps twice, right? So the option is 2. Okay, next one. The two strands of DNA are held together by. Okay, so you have studied the structure of DNA like this. It was right. And here both of these strands are held together by what? Okay, both of these strands are held together by the nitrogenous base pairs which are present inside. But how these nitrogenous base pairing has been taken place? Okay, they are bound with the help of what? Which bonds? Option A, peptide bond. Option B, phosphodiester bond. Option C, hydrogen bonds. And option D, disulfide bonds. As you already know, the two strands, okay, so the two strands of DNA are held together by, always you know that between two strands of the DNA, okay, between any two strands of the DNA, there are the presence of hydrogen bonds between the nitrogenous base pairs of one particular polynucleotide chain and the other polynucleotide chain. Okay, between these two, I'm drawing here. Okay, you can go through this one. So, between these two, what will be there? The nitrogenous bases will be present here, and these nitrogenous base pairs, okay, have been attached with one another with the help of what? The hydrogen bonds. Okay, these are nothing but what? The hydrogen bonds. And with the help of hydrogen bonds, both of these polynucleotide chains are held together. So coming to the next question, gene is, okay, gene is, what is gene? Option A, it is a segment on DNA. It is a segment of chromosome. It's a functional unit of the DNA, and it is the segment that is capable of crossing over. Okay. Gene is simply, you can say that it is a segment of DNA. Okay. Gene is a segment of DNA. Okay. So, next one, nucleotide arrangement in the DNA can be seen by. Okay, nucleotide arrangement in the DNA can be seen by option A, X-ray crystallography, option B, electron microscope, option C, centrifuge, and option D, light microscope. See, regularly whatever we are going to use in our laboratory, that will be what? Simple or compound microscope, which will be in its higher version will be what? A light microscope. Centrifugation is used to separate the particles, okay, heavier particles and lighter particles, that we are going to use the centrifuge. And electron microscope, which was used to take the photograph of what? The chromatin material, that means nucleosomes. But nucleotide arrangement is seen with the help of what? The X-ray crystallography. Okay, nucleotide arrangement will be seen by the help of X-ray crystallography. Clear? Coming to the next question. Nucleic acids are made up of, okay, nucleic acids, that means the DNA and RNA, they are made up of dash. Option A, nucleotides. Option B, nucleosides. Option C, amino acids. And option D, proteins. You know that nucleic acids are made up of what? Option A, nucleotides. Nucleotides are the building blocks of nucleic acids. And you know what is the difference between nucleotides and nucleosides? Okay, nucleosides will have only two units. Okay, they are the pentose sugar and nitrogenous base. But nucleotides will have three units. Okay, pentose sugar, nitrogenous base, and phosphate group. Right. So if all the three are there, then a nucleic acid can be formed. That is why nucleic acids are made up of nucleotides. Coming to the next question. Hydrogen bonds between cytosine and guanine are. Okay. Hydrogen bonds between cytosine and guanine are. Okay. Option A 1, option B 2, option C 3, option D 4. That means they are asking the number of bonds present between, number of hydrogen bonds present between the cytosine and guanine. You already know that cytosine and guanine will be bound with each other with the help of what? 3 hydrogen bonds. It is 3 hydrogen bonds. Okay, and at the same time, you can also recall that adenosine and thymine are bound up with the help of what? 2 hydrogen bonds. Adenosine and thymine are bound with the help of 2 hydrogen bonds, and cytosine and guanine are bound with the help of what? 3 hydrogen bonds. Next, coming to the 11th question. What is true for Watson and Crick's model of DNA? It's duplex width. It is a duplex width. Option A 10 pairs and 10 base pairs and 34 angstrom distance for every turn. Option B 10 base pairs and 3.4 angstrom distance for each turn of the spiral. Option C 20 base pairs and 31 angstrom for each turn. Option D none of the above. Okay, here you can directly take off, okay, the option C. Option C can be discarded very easily. Why? Because in the option C they have given 20 base pairs will be there for each turn. So it is not possible to have 20 base pairs for each turn. That is why option C is deleted. Then option A and B, if you observe very clearly, in the option A or option B, okay, take the option B first. 10 base pairs and 3.4 angstrom they have mentioned. If it is 3.4, then angstrom will not be proper here. If they have given nanometer, then the option B could have been the proper answer, but they have mentioned angstrom. That is why option B is also wrong one, right? Then what about option A? 10 base pair and 34 angstrom distance for every turn. Yes, it is proper one. So option A. In the DNA, okay, if you observe one complete turn, see, I am drawing here, in one complete turn of the DNA. One complete turn means from this particular point here, I am showing you from this particular point to this particular point. Okay, it makes one complete turn. In this one complete turn of DNA, what will be the length of the DNA? It will be 34 angstrom. Okay, so this length will be what? 34 angstrom. Okay, 34 angstrom. And how many base pairs will be there in this 34 angstrom length of the DNA? Okay, it will be having 10 base pairs. So option A is the correct answer. Clear? Then option, okay, coming to the next question, that is the 12th one. Nucleotide base present in the DNA and not in the RNA. Okay, you know already there are 4 nucleotides present in DNA and 4 nucleotides present in the RNA. Out of these 4 nucleotides which are present in DNA and RNA, okay, the difference occurs between DNA RNA in only one nitrogenous base or nucleotide. So in case of DNA you can find adenine, then second one is guanine, then cytosine, then thymine. Okay, adenine, guanine, cytosine, and thymine you can see. In case of RNA, okay, in case of RNA, whatever you can see, here adenine will be same or common between both DNA and RNA, and guanine will also be the same, and then cytosine will be same again, but instead of thymine, okay, instead of thymine, in the RNA, what will be there? Uracil will be there. Okay. So if you observe these 4, okay, the question is, see, nucleotide base present in DNA, not in RNA. Okay. So adenine, guanine and cytosine, they are present in DNA as well as RNA. But thymine is present in DNA, not in the RNA. Right? So thymine is present in DNA and not in the RNA. That is why option C, thymine, is the answer. Right. So coming to the next question, that is the pyrimidine base present in RNA in the place of thymine of the DNA is, okay, pyrimidine base present in the RNA in the place of thymine of the DNA is. That means the same question is asked in otherwise or other manner. Okay. So whichever is present in the RNA but not the DNA. Okay, that question is asked in the 13th one. So in the RNA, what is present? Which pyrimidine base is present? Uracil is present instead of what? Thymine. Okay, instead of thymine in the RNA, uracil is present. Clear? Coming to the next question. Which is true according to Chargaff's rule? Okay. So Chargaff, what he said, in a double helical model of DNA, okay, in a double helical model of beta DNA or in the double helical model of DNA, what will be there? The amount of purines is equal to amount of pyrimidines. Okay, the amount of purines will be equal to amount of pyrimidines. Or else, he also said, or in other way, it can also be said that the ratio between purines and pyrimidines will be equal to 1 or it will be constant. So here, according to Chargaff's rule, which of the option will be correct? So directly you can take off the option B and option C because they are completely wrong. Is that clear? Because you already know that adenosine binds with thymine and guanosine binds with cytosine. Here they have given it wrong. That is why option B and C are taken off. They are wrong. So look at option A and option D. Okay. In the option D, they have mentioned it in the ratio form, and we say that the ratio of purines will be equal to pyrimidines, or ratio of purines and pyrimidines will be equal to 1. Here they have given the ratio of purine and pyrimidine, okay, but they have not mentioned here the sum of purines and sum of pyrimidines properly. Okay, they have added A plus T. Actually, it should be A plus G and C plus T. Okay, actually, it should be what? A plus G by C plus T. But here what they have given? A plus T they have given. They have given the purine and pyrimidine. That is why option D is also wrong. So the right option is what? Option A, that is the sum of purines will be equal to sum of pyrimidines. Directly it is there. Sum of purines. Which are the purines? Adenosine and guanosine. Which are the pyrimidines? Cytosine and thymine. Okay, the sum of purines and pyrimidines will be equal. That is option A. Okay, according to Chargaff's rule, the sum of purines and pyrimidines will be equal. Right? So coming to the next question. In AGCT, that means in adenosine, guanosine, cytosine and thymine of the DNA, hydrogen bonds and base pairing occurs between. Okay, hydrogen bonds and base pairing occurs between. In these particular nitrogenous bases, okay, the pairing occurs between option A, adenosine, guanosine, cytosine and thymine. Option B, adenosine and cytosine, guanosine and thymine. Option C, adenosine and thymine, cytosine and guanine. Option D, all of the above. As you already know in the previous question itself, adenosine always binds with what? Thymine with the two hydrogen bonds, and guanosine always binds with cytosine with the three hydrogen bonds. Right? That means pairing will take place between adenosine and thymine and cytosine and guanine, never between others. Okay? Okay? So here pairing will be always taking place between what? Adenosine and thymine. Okay? Or else thymine and adenosine or vice versa. Okay? At the same time, guanosine and cytosine and vice versa. Right? So the option C will be the correct answer here. The pairing will be taking place between adenosine and thymine and cytosine and guanosine. Right, coming to the next question. Nucleotide pairs present in one turn of the DNA helix. Okay, nucleotide pairs present in, see here I am drawing the DNA helix. Okay, so this is DNA helix. In this DNA helix, how many nucleotide turns are, or how many nucleotide pairs are present here? In one complete turn of beta DNA, as we have already discussed earlier, how many base pairs will be there? So 10 base pairs will be present. Okay, in one beta DNA, in one complete turn, what will be there? 10 base pairs will be present. So the option here is option C, that is 10. So coming to the next question, nucleic acids were discovered by. Okay, nucleic acids were discovered by. It is the first point that you come across when you start studying this point or this particular chapter. Okay, nucleic acid. The DNA was discovered. Okay, by Mischer in the form of what? Nuclein. Okay. Mischer I identified the DNA or genetic material and named it as what? The nuclein. So you know or you remember that point, I hope. Okay. So according to that, the answer will be what? The Mischer. Okay. Mischer identified the DNA for the first time and he did not know that it is DNA. That is why he had given the name nuclein for the DNA. Later on, okay, the nuclein was completely deciphered by other scientists and given with the name that is DNA. Right, coming to the next question, that is the type of coiling in the DNA is. Okay, the type of coiling in the DNA is option A, right handed side, option B, left handed side, option C, zigzag, and option D, opposite. As I have already discussed this in the theory class, DNA will be double helical model or double helical structure in which both the polynucleotide strands, okay, or polynucleotide chains are running in anti-parallel direction to each other, which are right-handedly coiled. Okay, as a direct question they have given, and by the explanation which I have given, directly you can answer. Okay, DNA is double helical model made up of two polynucleotide chains that run in anti-parallel direction to each other and are coiled in right-handed manner. I have told you, right? So the answer here is option A, right-handed. Right? It is not left-handed or it is not zigzag or opposite. Right? Coming to the next question, that is 19th question. Length of one coil of beta DNA helix is, okay, that means one complete turn of beta DNA helix, what will be the length? It is 3.4 nanometer or 34 angstrom. 3.4 nanometer or else 34 angstrom. Then what about this 0.34 nanometer? It is the length between any two base pairs. But here what they have asked the question? A total length of one coil. So that will be equal to what? 3.4 nanometer. Right? So coming to the next question. Purines of DNA are, okay, as we have already discussed in the theory part, the polynucleotide chains, okay, structure of polynucleotide chains. So it will be having what? Nitrogenous base pairs or nitrogenous bases. How many types of nitrogenous bases are there? Two types, purines and pyrimidines. And what are purines? Double ringed structures which are adenosine and guanosine by their name. Okay, and what about pyrimidines? Single ringed structures which are three in nature. They are the cytosine, thymine, and uridine. Right. Here they are asking about what? The purines. The purines are nothing but adenosine and guanosine or adenine and guanine. Right. So in the option B, both are properly mentioned. That is why option B is right answer. Adenine and guanine. Nucleoside. Coming to the next question, that is nucleoside. So what is nucleoside? Option A, nitrogenous base and phosphate group. Option B, phosphate group and sugar. Option C, nitrogenous base and sugar. Option D, nitrogenous base, sugar and phosphate. See, you already know that nucleotide and nucleoside, there is a difference of 1 unit. In case of nucleotide, okay, in case of nucleotide, all the three will be present, all the 3 units, that is nitrogenous base, pentose sugar and phosphate group. But in case of nucleoside, okay, the phosphate group will be absent. If the phosphate group is absent, then which all the remaining part will be there? Only nitrogenous base and pentose sugar. So option C will be the proper answer. Nitrogenous base and pentose sugar will be there in the nucleoside. Clear? So coming to the next question. Chargaff's rules are applicable to. Chargaff rules are applicable to what? Option A, single stranded RNA. Option B, single stranded DNA and RNA. Option C, single stranded DNA. Option D, double stranded DNA. Always do remember, Chargaff's rule is applicable only for, okay, double stranded molecule, that is DNA, not the single stranded molecule. Okay, Chargaff rule is applicable only for the double stranded molecule but not the single stranded molecule, right? That is why the option, in only one option they have mentioned double stranded, that is option D. So it is the proper answer here. Okay, so Chargaff's rules are applicable only for double stranded DNA. Okay, it is applicable only for the double stranded DNA, right? Next question. Who wrote, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." Okay, when the scientists were discovering, or when the scientists were discussing about the replication method of the DNA, okay, the replication method of the DNA, when they were discussing, then this particular postulate or this particular sentence or this particular point was given. Okay, so it was given by option A, Michelson and Stahl. Option B, Beadle and Tatum. Option C, Watson and Crick. Option C, Nirenberg and Ocha. Right here, the semi-conservative method of DNA replication or the type of DNA replication was first described by whom? The Watson and Crick. Even though these two are the same scientists who have given the structure of DNA, double helical model of DNA, and the same scientists have given what? This particular idea of semi-conservative method of DNA replication, right? And what about Michelson and Stahl? They have given experimental proof that DNA will undergo semi-conservative type of DNA replication only. Okay, the experimental proof was given by Michelson and Stahl here, but the type of DNA replication is semi-conservative was told by Watson and Crick. Okay, that is why this particular sentence was told by whom? Watson and Crick. So the correct answer is C. Is that clear? Then coming to the next one. Length of the DNA with 23 base pair is, okay, length of the DNA with 23 base pair. In the theory part, when I was discussing with this particular structure of DNA, I had told you in the NEET examination or in the CET examination, they may ask you this type of questions to calculate the length of the DNA. How can you calculate the length of the DNA? Very simple. Number of base pairs, okay, should be multiplied with, okay, number of base pairs should be multiplied with what? Distance between any two base pairs. Okay, so distance between any two base pairs is, as you already know, it is 0.34 nanometer. Okay, 0.34 nanometer or else 3.4 angstrom. Okay, here they have asked in the form of what? Angstrom. So let us do that. Okay, length of the DNA with 23 base pairs is, that is 3.4 angstrom multiplied with How many base pairs are there? 23 base pairs are there. So the answer will be 78.2 angstroms. Okay, 78.2 angstroms. So option B is proper one here. Okay, just you have to multiply number of base pairs with what? Distance between any two base pairs. So here number of base pairs is 23. So 23 should be multiplied with what? Okay, 3.4 angstrom, because the distance between any two base pairs is what? 3.4 angstrom. Okay, since the answers are given in angstroms form, you can use 3.4 angstrom. If it is asked in nanometer form or nanometer things, you can multiply with what? 0.34 nanometer. Is that clear? So depending upon what measurement they have given, upon that you have to use it. Clear? So next question. DNA strands are anti-parallel because of. DNA strands are anti-parallel because of dash. See, first of all, DNA strands are formed with the help of, okay, long chain of what? Okay, the DNA strands are formed with long chain of polypeptides. Yes or no? So these polypeptides are formed with the help of what? Okay, the phosphodiester bonds. Okay, the polypeptides, okay, polypeptide chains are formed by phosphodiester bond. Due to presence of phosphodiester bond or due to occurrence of phosphodiester bond between the nucleotides, what will happen? The DNA strands will be running in anti-parallel direction to each other. That means one will be running from 5 dash to 3 dash direction and the another one will be running from 3 dash to 5 dash direction. Right? Next question. In the DNA segment of 6 coils, okay, in the DNA segment of 6 coils, 22 base pairs are linked by 2 hydrogen bonds. Okay, see, a bit applied question is there, but the easiest one. In DNA segment of 6 coils, 22 base pairs are linked by 2 hydrogen bonds, and how many cytosine base pairs would be there, or how many cytosine bases would be present? So what they have given, the DNA segment is there with 6 coils. 6 coils means a total of how many base pairs will be there? 60 base pairs. Okay, because in each coil, each complete coil will be containing 10 base pairs. So total number of base pairs in 6 coil will be what? 60. Right? And out of these 60, 22 are what? Bound with 2 hydrogen bonds. That means adenosine and thymine, as you already know, they will be bound with, okay, adenosine and thymine will be bound with 2 hydrogen bonds. A binds with T with the 2 hydrogen bonds, as you already know. And remaining part is what? Okay, 22 adenosine and thymine are there. Indirectly they have given there are 22 adenosine and thymine. If out of 60, 22 adenosine and thymine are there, then what about the cytosine base pairs? Just take off 22 from 60. Then the remaining will be what? 38. So 38 cytosine bases would be there. Is that clear? ಅಂದರೆ 60ರಲ್ಲಿ 22 ಏನಿದಾವು ಅಡಿನೋಸಿನ್ಸ್ ಆ್ಯಂಡ್ ಥೈಮಿನ್ಸ್ ಇದ್ದರೆ ಓಕೆ ಉಳಿದಿದ್ದೆಷ್ಟು ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಅದರಲ್ಲಿ 60 minus 22, that will be equal to 38. So 38 cytosine bases would be there. Right? Next question. Information flow or central dogma of modern biology, or else it is central dogma of molecular biology, you can call it. Okay? Information flow or central dogma of molecular biology is dash. Okay, do remember that central dogma of molecular biology is nothing but the process of, okay, transcription and translation. Okay, the process of transcription and translation itself is called as central dogma of molecular biology. Okay, always how exactly it will be taking place? It will be taking place from DNA. That means the information flow will take place from DNA to RNA and RNA to proteins. So in the options where the DNA, RNA and proteins is there, in second option, see here, information flow will be first taking place from DNA to RNA. That means the information present in the DNA will be first converted into RNA, or first will be formed as RNA. Okay, then that information which was taken from DNA as RNA will give rise to proteins. That means DNA, RNA and proteins. This is the way how exactly the information flow will take place. Clear? The next question, which one is not true? Which one is not true? Option A, adenosine binds with thymine and guanosine binds with cytosine. Here see, adenosine binds with thymine, it is correct. That too with the 2 hydrogen bonds, it is perfect. Then guanosine binds with the cytosine with the 3 hydrogen bonds, it is correct. But what they have asked, which is not correct. Okay, so option A is correct here. What about option B? Okay, adenosine binds with guanosine, no, it is wrong. Adenosine binds with thymine, right? Then this is not true, this is not true. That means this is the correct answer here. But let us confirm about C and D even. In the C, they have given cytosine binds with guanosine with 3 hydrogen bonds. Yes, perfect. Then thymine binds with adenosine with 2 hydrogen bonds. Yes, it is also correct. Then what about C? What about option D? Guanosine binds with cytosine with 3 hydrogen bonds and adenosine binds with thymine with the 2 hydrogen bonds. It is also correct. So option A, C and D are properly mentioned here, and option B is wrong. So that is why, what is there? The option B is not true. Okay, option B is not true. Next question, which is false? Okay, out of these 4 options, which is false? Option A by T is 1. That means the ratio of A and T will be equal to 1. Okay, of course, adenosine always binds with thymine. That is why number of adenosines will be always equal to number of thymine. So if you take the ratio of A and T, it will be always equal to 1. That is correct. Option A is correct here. And what about option B? A plus G is equal to C plus T. That is also perfect. That is, the purines will be equal to pyrimidines. Option A and B are correct only. But what they are asking? They are asking the wrong one. Then option C is also correct. Okay, C by G, that means cytosine by guanosine, they will be equal to 1. Yes, of course it is correct. If option A, B and C are correct, then look at the option D. A plus T will be equal to C plus G. This is not correct. So the option is D. Why? Because A plus G and C plus T will be equal. Okay, but not A plus T and C plus G. Clear? Because the ratio of, or else the sum of purines will be equal to pyrimidines. Okay, sum of purines will be equal to pyrimidines, but not sum of one purine and pyrimidine equal to another purine and another pyrimidine. Is that clear? That is why option D is wrong and it is false here. According to the question, we have to identify the false one. That is why option D is false. Right. Next question. Adenine is 30%. What would be the percentage of guanine? Okay. Adenine is 30% and what would be the percentage of guanine? Very simple. If they ask in the form of percentage, okay, what you have to do? Just take, okay, percentage means always we will consider it for what? 100. Okay, 100%. In 100%, adenine is what? Okay, in 100%, adenine is, okay, 30%. Okay, in 100, adenine is 30%. And if adenine is 30%, then it will be equal to thymine. So thymine will be what? 30% again, because you already know the adenine will be always equal to thymine. So in the DNA, if 30% of adenine is there, then 30% of what will be there? Thymine will be there. So how much is remaining? So remaining will be 40%. Okay, 100 minus 60, that will be equal to what? 40%. Right? In this 40%, okay, who will be there? the guanine and cytosine will be there. Okay, in this 40%, guanine and cytosine, if you distribute equally, then it will be 20% of guanine and 20% of cytosine will be there. Okay, 20% of guanine and 20% of cytosine, right? So here what they have asked, adenine is 30% and what would be the percentage of guanine? Okay, now you got the percentage of guanine, that is equal to what? 20%. So option B, 20% is correct. Okay, if you ask, okay, if you are asked with the percentage, okay, then you have to do it like this. Okay, if you are asked with the question of percentage or in the form of percentage, you have to calculate the percentage of the bases like this. Okay, next question, DNA of certain organisms has guanine in 20 of the bases. Okay, in 20 bases what is there? The guanine is there. What would be the percentage of bases? Would be adenine. How many percent of adenine will be there? Okay, very simple. The reverse question will be asked here, or reverse question is asked. ಅಂದ್ರೆ ಮೇಲ್ಗಡೆ ಕೊಟ್ಟಿದ್ದು, they have asked adenine is 30%, what is the percentage of guanine? And here they have given the guanine, that is 20. And what would be the percentage of adenine? Very simple, it is 30. Is that clear? Right? Next one. If there are 120 adenine molecules in beta DNA molecule or beta double helix DNA showing 20 coils, what is the pyrimidine nucleotide forming 3 hydrogen bonds? See, simply they have asked, okay, what they have asked, if there are 120 adenine molecules, so adenine molecules, A is equal to 120, okay, A is equal to what? 120 adenine molecules are there, right? Let us consider 120 adenine molecules separately here. And where the 120 adenine molecules are there? In the beta DNA double helical structure showing 20 coils. 20 coils means total number of base pairs is equal to what? 200, because each coil will be containing 10 base pairs. So if there are 20 coils, then 200 base pairs will be present. Out of 200 base pairs, okay, out of 200 base pairs, adenine is 120. Okay, adenine is 120. If adenine is 120, then what will be there? Cytosine will be always equal to adenine, that is 120. Okay, cytosine will be 120 again, and remaining is what? 80. Okay, that will be the pyrimidine nucleotide that form 3 hydrogen bonds. So A and T will form 2 hydrogen bonds, that will be equal to 120. And if you take off 200 from, 120 from 200, remaining will be what? 80. Is that clear? So that will be the number of nucleotides forming 3 hydrogen bonds. That means they have asked about what? The guanine and cytosine. If out of 200, 120 is adenosine and thymine, then what will be the number of cytosine and guanine? It will be 80. Is that clear? So next one, the process of multiplication of DNA from DNA is known as, okay, the process of multiplication of DNA from DNA. As you already know, DNA formation of, formation of DNA from a parental DNA is nothing but what? DNA replication. Directly you can answer. And what is the DNA duplication? Okay, what is the difference between replication and duplication? Do remember that in the replication, complete molecule of DNA will be copied. Complete molecule. That means after replication, 2 daughter DNA molecules can be formed. But duplication, okay, refers to what? The copying of DNA in only one particular part, or only a part of DNA will be copied or duplicated. Okay, not the complete replication. Is that clear? Next one, transcription. As you already know, it is the process of formation of mRNA from DNA. Okay, formation of or biosynthesis of mRNA from DNA is transcription. And what about translation? The process of formation of protein from the mRNA. Okay, the process of formation of protein from the mRNA is called as translation. Right? Next question. The area of unwinding and separation of the DNA strands during replication is called. Okay, the area of unwinding and separation. Where exactly the DNA replication begins. Okay, the DNA replication begins at a particular point called as ORI site, as you already know, and that ORI site itself is called as origin of replication, site of origin of replication. Is that clear? That is ORI site, where exactly unwinding and separation of the DNA strands will be initiated. Clear? Then topoisomerase is involved in. Okay, topoisomerase is involved in. Okay, option A, producing RNA primer. Option B, joining of the DNA segments. Then option C, producing nick in the DNA. Option D, separation of the DNA strands. As you already know, the DNA helicase enzyme is also called as topoisomerase. Okay, and what is the function of DNA uh, helicase enzyme? The DNA helicase enzyme help for the separation of the DNA strands. Okay, that is the function of topoisomerase, because topoisomerase is another name of what? DNA helicase. Right. Then what about the enzyme that help for joining of the segment? It is DNA ligase. Okay. Next one. In DNA replication, the primer is, okay, in DNA replication, primer is, what is primer? Okay, option A, a small deoxyribonucleotide polymer, okay, or else a small DNA fragment they have given. Option B, small ribonucleotide polymer, or else a small RNA fragment. Option C, helix destabilizing protein. Option D, enzyme that take part in joining the nucleotides to their complementary base. Is that clear? But here, the DNA replication, during the DNA replication, the primer is what? A small ribonucleotide, because it is a small segment of RNA. Okay, the primer in case of DNA replication will be what? RNA primer. You already know that during the DNA replication, the RNA primers are used. Clear? Next one. DNA strand is synthesized in the direction. Okay, DNA strand is synthesized in the direction. Always you know that synthesis of DNA will be taking place from 5 dash to 3 dash. Okay, okay, always the synthesis of DNA, always the formation of new strands of the DNA will be taking place in the direction of 5 prime to 3 prime direction, or 5 dash to 3 dash direction. Okay. Next, if the question is asked in another way, that is, the DNA synthesis takes place in the direction of the template. Okay, if they give the name, or if they ask about the template's direction, then you have to write 3 dash to 5 dash. Okay, if they ask directly without any mentioning of template, then you can directly go for what? 5 dash to 3 dash, because formation of new strand of the DNA will be always taking place from 5 dash to 3 dash direction. Okay, if the formation should be taking place in the 5 dash to 3 dash direction, then the template should be in the form of what? 3 dash to 5 dash. That is why if they ask you about the template, then you go for 3 dash to 5 dash direction. Here they have not mentioned about any of the template, that is why the answer will be 5 prime to 3 prime, or 5 dash to 3 dash. Right? Coming to the next one, Okazaki segments are, or Okazaki fragments are. What are Okazaki fragments? Option A, small segments of RNA. Option B, small peptides. Option C, small DNA segments. Then option D, small DNA segments formed over DNA template running in 5 dash to 3 dash direction. Okay. So which one is the correct answer? Okazaki fragments. As you already know, Okazaki fragments are the smaller DNA segments. Okay, smaller DNA segments. Where they are formed? They are formed on the lagging strand. Okay, they are formed on the lagging strand. And what is the lagging strand? The strand of the DNA which will be having the polarity 5 prime to 3 prime. So here the Okazaki fragments are what? Small DNA segments formed over the DNA template running in 5 dash to 3 dash direction. If the template is running in 5 dash to 3 dash direction, then continuous synthesis of new strand cannot take place. Why? Because the synthesis of new strand will be taking place in what? 5 dash to 3 dash direction, as we have discussed in the earlier question. Okay. If the template strand is also having 5 dash to 3 dash direction, then continuous synthesis cannot take place. Only if the template has got 3 dash to 5 dash polarity, on that continuous synthesis can take place. That is why here the option D is correct. Okazaki fragments are the fragments of the DNA that are formed over DNA template running from 5 prime to 3 prime direction. Right. Next one, Okazaki fragments are joined by. Okay. The joining of Okazaki fragments, because they will be the smaller fragments. Okay. These smaller fragments cannot be left as it is. They should be joined end to end. So the joining of the Okazaki fragments will be taking place with the help of what? Okay, option A, DNA polymerase, option B, DNA ligase, option C, DNA polymerase, then DNA polymerase 2 and DNA polymerase 1, option D. So here, as you already know, the polymerase enzymes help for polymerization, but not the ligation or attachment. Okay, here directly the answer is given DNA ligase, which help for ligation. Ligation means what? Attachment. Clear? Attachment. For the attachment or for joining of the Okazaki fragment, who will be helping? The DNA ligase will be helping. Clear? Next question. Okazaki fragments give rise to. Okay? Okazaki fragments give rise to dash. Option A, master strand. Option B, sense strand. Option C, lagging strand. Option D, leading strand. Okay? As I have already discussed in the 7th question itself, Okazaki fragments are joined, or sorry, 6th question itself. Okazaki fragments will be formed on what? Lagging strand. Is that clear? So that is why Okazaki fragments can give rise to what? The lagging strand. Is that clear? Okazaki fragments can give rise to lagging strand. Next one, 9th question. Leading strand during DNA replication is formed dash. By the name itself, you can understand leading strand. That means it will be synthesized continuously. Okay? Since it is synthesized continuously, it will be called as leading strand. Okay? So the answer is option A, continuously. Leading strand during the DNA replication is formed continuously. Clear? The next question. Bacterium grown over a medium having radioactive sulfur incorporates radioactivity in. Okay. So here the bacterium grown over medium having radioactive sulfur, that means sulfur is present in the proteins. You already know sulfur is present in the proteins. So if the bacteria are grown in a medium containing radioactive sulfur, then the radioactivity will be found in what? The protein of the upcoming bacteria. Is that clear? So radioactivity, if it is given with the sulfur, and sulfur if it is incorporated in the organism, it will form the protein structure of that particular organism. If you give normal sulfur, then normal proteins can be formed. If it is radioactive in nature, then radioactive proteins will be formed. That is, radioactivity can be seen in the proteins if you grow the bacteria in the radioactive sulfur medium. Okay. Coming to the next question. So here the answer is proteins. Next question. What is true about oocyte? Okay. What is true about oocyte? Option A, one in all organisms. Option B, several in all organisms. Option C, one in eukaryotes and several in prokaryotes. Option D, one in prokaryotes and several in eukaryotes. Okay. So the number of oocytes differ in case of prokaryotes and eukaryotes. Usually, the oocyte will be only one in prokaryotes and it is several in the eukaryotes. That is why the option D is the answer here. Okay. In case of prokaryotes, they will have only one oocyte. But in the eukaryotes, what will be there? Several oocytes will be present. Okay. In the eukaryotes, around 400 to thousands of what oocytes can be seen, but in case of prokaryotes only one oocyte will be present, right? Next question. Enzyme required during DNA replication is, okay, the enzyme required during DNA replication is option A, DNA polymerase, option B, DNA ligase, option C, both A and B, then option D, sigma factor. See here, enzyme required during DNA replication. No doubt DNA replication requires DNA polymerase enzyme. But in the options they have given DNA polymerase, DNA ligase. Then in the option C they have given both A and B. So for the proper DNA replication we need both A and B. That means DNA polymerase as well as DNA ligase. Why it is so? Because as you already know, in this particular DNA replication, on one strand continuous synthesis will be taking place, but on the other hand, in the other strand what will be there? The fragments will be formed, Okazaki fragments will be formed, and for the joining of these Okazaki fragments what is required? Ligase is required. That is why for the DNA replication in a complete manner, what is required? Both DNA polymerase enzyme as well as DNA ligase enzyme are required, right? Next question. DNA polymerase helps in dash. Okay. DNA polymerase helps in option A, splitting of the two DNA strands. Option B, proofreading of the DNA. Option C, renaturation of the DNA. Option D, joining of monomers of the DNA. See, by the name itself you can understand polymerase. That means it helps for polymerization process. And what do you mean by polymerization? That means formation of the polymers. Okay. DNA polymerase enzyme help for formation of DNA polymers. Okay. If it help for the polymerization, then you can understand that it helps for attachment of several monomers. Okay. When several monomers are attached end to end, then it will make what? One polymer, right? So here DNA polymerase help for what? Joining of monomers of the DNA. Clear? Next question. Semi-conservative method of DNA replication using 15N was demonstrated by, okay, heavier isotope of nitrogen was used and the semi-conservative method of DNA replication was demonstrated by, okay, that means experimental proof of semi-conservative method of DNA replication was given by whom? Option A, Michelson alone. Option B, Taylor. Option C, Michelson and Stoll. Option D, Hershey and Chase. Okay, direct answer is there and direct question, that is semi-conservative method of DNA replication is given by whom? The Michelson and Stoll. Clear? Then, next question. DNA replication requires option A, DNA polymerase, option B, RNA polymerase and translocase, option C, DNA ligase, and option D, DNA polymerase and DNA ligase. Okay, I think it is a repeated question with different kinds of what? The options. Okay, same question with different aspect or different angle of what? The set of options. Okay, here the answer is DNA polymerase as well as DNA ligase. Next question. Ligase is an enzyme required for, okay, as you already know, ligase help for joining. Let us go through the options. Option A, breaking of the DNA, no. Option C, option B, joining of DNA bits, of course, yes. Okay, so the option B is what? Proper answer here. And what about option C, renaturation of the DNA? Option D, proofreading, no. Option B, joining of the DNA bits. Okay, what do you mean by DNA bits here? DNA fragments, right? Next question. DNA duplication or multiplication is known as, okay, DNA duplication or multiplication is known as DNA replication. Okay, as you already know, DNA replication it can be called as. But friends, do you remember? Okay, you have to remember that duplication and multiplication, okay, according to this question they are referring it as replication, but duplication cannot be same as that of replication. Duplication means only a part will be, okay, copied. Multiplication holds good for what? Replication. That is why the answer will be replication, option A. Actually, the word duplication should not be used here, but since I want to make you know that there is a difference, okay, I have added it here. Clear, right?